Bismillahirrahmanirrahim. Today's topic is on modes of inheritance of genetic disorders. The book does not include properly all those modes, but they are randomly distributed. So, in next 4 to 5 pages, we will learn about them. So, first of all, we have to categorize them. Here on page 56, I have written the types. The first type is single gene disorder with classical Mendelian inheritance. We know them. Gregor Mendel did experiment, and those experiments yield a few scientific patterns of inheritance. They were autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and the final one was of codominance pattern. So here we will see all these patterns in humans. And the next is single gene disorder with non-classical or non-Mendelian transmission pattern. So they can also be called as non-classical inheritance disorders, but of single gene. They are trinucleotide repeat disorders like Huntington disease, myotonic dystrophy, Fragile X syndrome, and Friedreich ataxia. So Fragile X syndrome is also here and also in X-linked disorder because it is of X-linked disorder, Fragile X chromosome, but because it is a trinucleotide repeat expansion, so it's also included in this other category, that is single gene disorder with non-classical inheritance. So the other non-classical inheritance is mitochondrial inheritance patterns. We will see Mila's syndrome in it, famous example asked often. Then genomic imprinting disorder. There are two disorders given on this page: Prader-Willi and Angelman syndrome, both related to chromosome 15. Both are important. And then moving on to multi-gene or polygenic disorder, and sometimes or most of the time involves the environmental factors, so it is multifactorial disorder. They are the most common overall medical disorders, uh, including diabetes mellitus, hypertension, congenital heart disease. So these examples are asked, or in otherwise multifactorial disorders are most common. And then comes the single gene disorder, autosomal recessive types, second most common. And then comes the chromosomal disorders. Chromosomal disorders are either structural disorder like deletions, insertion, inversions. These are not much asked, but numerical disorders either with autosomal or with sex chromosomes are asked. Autosomal numerical disorder or cytogenetic disorder in other language are mostly trisomies. One extra chromosomes is mostly from meiotic non-distinction uh, method. So whenever there is meiosis, chromosome do not divide centrally and one chromosome goes extra in one cell, then there it is a phenomena called trisomy, and one cell won't have one chromosome uh, or one less chromosome. So mostly it is the cell or the egg with extra chromosome, it is fertilized. So it is trisomy 21 either, or 18 or 13 different syndrome. Down syndrome is most common trisomy, and it is often asked. Then sex chromosomal disorders involves X or Y chromosome. If one extra X is there, it is called Klinefelter, and it is most common sex chromosomal disorder. It is also asked. And if X is deleted or not there, then it is Turner syndrome. So it is discussed on page 638 with sex disorders, because these both are sex disorders, so we won't discuss much them here. We'll discuss trisomies. uh, just finishing this page, we have a single gene disorder with non-classical inheritance. They are three types, and third, genomic imprinting disorders are given here. There are two types, either the Prader-Willi syndrome or hyper obesity syndrome, because in this there is hyperphagia which leads to obesity, and this is hyper obesity syndrome, asked in a different way. And it Prader-Willi involves the paternal gene, so it is paternally deleted or mutated gene, that is also asked, P for paternal. And Angelman has M in it, it is a maternally deleted or mutated allele, and the most important symptom is inappropriate laughter or happy puppet. So these obesity and happy puppet and maternal and paternal inheritance are asked. We'll complete these single gene disorder with non-classical inheritance here because they are less details about them. Here it is modes in under modes of inheritance. Instead of fifth codominance pattern, they have written mitochondrial inheritance. It is in fact single gene non-classical inheritance. We know that the sperm tail has mitochondria, and while penetrating egg it loses the tail outside, and it is only the genetic material that is transmitted. So by this, the mitochondria of all human species or whole human race is only being transmitted through mothers. So mitochondrial DNA is transmitted through mother, one MCQ, and mitochondrial inheritance is through mother, other MCQ. And important disease is Mila syndrome, that is mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. We know that mitochondria are abundant in muscles, so it is myopathy. So muscle biopsy will show rigid red fibers. These are basically having accumulation of diseased mitochondria. So it is transmitted only through mothers. And the third type is trinucleotide repeat disorder. These are Huntington disease, myotonic dystrophy, Fragile X syndrome. Most of them are also included in single gene disorder with classical inheritance pattern, but they have three nucleotide repeated in a sequence, so that's why they are in subcategory of trinucleotide repeat disorder. Otherwise they are either autosomal dominant, like Huntington disease is autosomal dominant, or Fragile X syndrome is an X-linked recessive disorder in its true sense. So these three and four examples are discussed here on page 62. The Fragile X syndrome is most common inherited cause of intellectual disability, but Down syndrome is most common genetic cause. So genetic cause is asked Down syndrome. Second most common genetic is Fragile X syndrome for intellectual disability. And if the word inherited is used, inherited means it can be inherited from one to another, but it is not genetic, not necessarily from genes. So these mutations in Fragile X syndrome has multiple reasons. One, mostly it is the CGG sequence repetition that is leading to Fragile X syndrome, and it is X-linked recessive, we know that. And the other is Huntington disease, CAG repetition. It is an autosomal dominant disease and often asked in MCQs. Uh, for Huntington disease, quadrate nucleus of brain is involved, that is also MCQ asked. So we have not to confuse. The Down syndrome is the most common genetic cause of intellectual disability, and second is Fragile X syndrome. And then the Fragile X syndrome and Huntington disease, although they have their own mode of inheritance, either autosomal dominant or X-linked dominant pattern for Fragile X syndrome, but in fact they are trinucleotide repeat expansions. So coming back to the point that it is an autosomal dominant Fragile X syndrome. If wrongly recessive was said before, that was not meant to be. It is autosomal dominant pattern. And now moving on to these single gene disorder with classical Mendelian inheritance, we have been studying them for long. These are autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and one is codominance pattern. So codominance pattern is discussed here, just two MCQs. Both the alleles are expressed, for example, HLA on WBCs, human leukocyte antigens, or blood group antigens, ABO antigens on RBCs. These are examples of codominance. Alpha 1 antitrypsin disease is also of codominance pattern, but these two physiological phenomena are of important from exam point of view. And then these single gene Mendelian disorders are important, and examples of each is on the next page: autosomal dominant, autosomal recessive, and then X-linked recessive, and then of course uh, X-linked dominant as well. So we'll go through them. First, their general pattern of inheritance, that is important. Autosomal dominant, we know that if 20 we have 23 pairs of chromosomes, so only 23rd is not involved, that is XY or XX. Other any chromosome, it is an autosomal. And autosomal dominant means if one gene is affected, then it will be transmitted to almost half of the next generation, and all one by two or 50% will be affected with one affected heterozygous parent. On average 50% children will be affected, as MCQ asked. And there is no carrier state that one carries. If one has the gene, it expresses in most of the cases, and if it does not express, it is called reduced penetrance. That is a phenomena associated with autosomal dominant. It is asked if a disease is not expressed because of certain reasons, but there is a defective gene in body, that is called reduced penetrance. And then we have or incomplete penetrance. And then variable expressivity is also associated with autosomal dominant pattern. It is the different severity of disease. For example, neurofibromatosis, NF, one can have fibromas in body 100, other patient can have 200, but he will have or he will not have. So it is associated with autosomal dominant. Variable expressivity, these MCQs are asked from autosomal dominant, and the examples are given in next page. They are also asked. We'll discuss examples with each pattern. So autosomal disorder, autosomal dominant disorder, we have achondroplasia. It will have short upper arms and lower limbs as well. Most common cause of short stature, genetic cause of short stature asked. Details on page 462. Then autosomal dominant polycystic kidney disease. We need to know that dominant is for adult type, and autosomal recessive polycystic kidney is for infantile or uh, non-adult type. So these are to be compared. Dominant, it is autosomal uh, uh, dominant polycystic kidney is in adults, and recessive in infantile. So infant case will be given, it will be autosomal recessive, and adult is given, it is autosomal dominant polycystic kidney disease. Then familial adenomatous polyposis, FAP, and familial hypercholesterolemia. Hypercholesterolemia is given on page 94. It is type 2 dis beta lipoproteinemia, and type 4 is also autosomal dominant, and other two types 1 and 3 are autosomal recessive type. So these all are asked, but important is familial hypercholesterolemia, like familial adenomatous polyposis. It is also autosomal dominant. Then hereditary hemorrhagic telangiectasia, not important. Then but hereditary spherocytosis, and mostly spectrin gene is spectrin protein is involved. It is also given in blood chapter. Huntington disease, we know that it is trinucleotide repeat expansion, but it is an autosomal dominant disease, and quadrate nucleus is involved. Then Marfan syndrome, fibrillin 1 gene, and long stature with ectopia lentis patient. We have short stature for achondroplasia and long stature for Marfan syndrome, and patient presents mostly with a case scenario of cystic medial necrosis and aortic rupture patient. So fibrillin 1 gene, long stature, long arm, and ectopia lentis, it is Marfan syndrome. These are mostly structural genes for autosomal dominant. Then multiple endonuclear neoplasm and myotonic muscular dystrophy, example of autosomal dominant. But Duchenne muscular dystrophy and Becker are X-linked recessive ones. These are important. Myotonic is dominant, and Duchenne muscular dystrophy is X-linked recessive. And then neurofibromatosis is important. It is an X-linked dominant. We know that variable expressivity is a phenomena associated with neurofibromatosis. And then both types of neurofibromatosis, then tuberous sclerosis, von Hippel-Lindau disease, and neurofibromatosis is called von Reckling-Ghosn disease type 1, and then von Willebrand. These three Vons are examples of autosomal dominant. Von Willebrand disease is autosomal dominant and most common cause of most common bleeding disorder, genetic bleeding disorder is also asked. Then from cancers, retinoblastoma is an autosomal dominant type is also asked. Not given here, you should add it up. Then autosomal recessive ones. Let's first discuss how they propagate. A recessive will only affect when both genes are in one individual, and one carrier cannot do this. Two carriers can transmit it. So two carriers, small a's, and only 1/4 will be affected. This is an MCQ asked. And most common Mendelian disorders are also autosomal recessive, and they involve mostly the enzymes. So enzymopathies, glycogen storage diseases, all are examples of autosomal recessive disorders. We will discuss here autosomal recessive on the next page. Albinism, because it is a pigment and it involves synthesis with enzymes. Autosomal recessive polycystic kidney disease with comparison to dominant is an the phenomena associated with adult, recessive with infantile uh, polycystic kidney disease. And then cystic fibrosis is important thing. We'll discuss in detail down. 2-3 MCQs are asked. And glycogen storage diseases, we know hemochromatosis involves enzymes, iron overload, and Cartagena syndrome is ciliary dysfunction. It is a structural disorder, although a bit, but it is included in recessive disorder. This is to be differentiated. Otherwise, neurofibromatosis and Marfan syndrome are structural disorder. They are primarily in dominant disorder. Mucopolysaccharidosis, again, except Hunter syndrome, they are enzymopathies, so recessive. Phenylketonuria, again enzymopathy, recessive. Sickle cell anemia, now coming to the blood disorders, sickle cell anemia and thalassemia are autosomal recessive disorder. And for thalassemia, and we have alpha thalassemia, chromosome number 16 deletion mutation, beta thalassemia due to point mutation in chromosome number 11. So in blood chapter they are discussed in detail, but this is important that chromosome number 16 for alpha, 11 for beta. And then Wilson disease, again copper metabolism, just like hemochromatosis, iron metabolism. This is also autosomal recessive. And cystic fibrosis, autosomal recessive, CFTR gene on chromosome number 7, and that is the transmembrane regulator for chloride channel. It is asked in GIT and lungs. So if chloride is not reabsorbed, there will be more not reabsorbed from sweat. It will lead to increased chloride secretion in the sweat, and it's a diagnostic test. Increased chloride secretion in cystic fibrosis is diagnostic test is asked in MCQ, and less chloride is secreted into the uh, lungs and GIT. It will leads to the thick mucus, and thick mucus will lead to recurrent pulmonary infection like staph aureus initial stage, P. aeruginosa in later on, and then pancreatic insufficiency, because pancreatic secretion won't be secreted, malabsorption and steatoria. So a clinical scenario of all these symptoms is asked for cystic fibrosis, and autosomal recessive is asked, or the CFTR gene on chromosome 17 is asked. Nasal polyps is also associated with cystic fibrosis. Detail is given here, but these are the MCQ. Increased chloride concentration in sweat gland is diagnostic for cystic fibrosis. Then next type is X-linked recessive disorders. We know that just like autosomal recessive, if a disease is X-linked recessive in females who have two X chromosomes, it will not be fully expressed unless both are affected. One, so female should have both affected genes. It means that it is more amongst male because only one X chromosome is present in male, and if that is affected, it will behave like a dominant trait, and that's why male is hemizygous for X-linked disorders. It is MCQ asks, male is hemizygous, not hetero. It is hemizygous because one X chromosome is already absent. Hemi means half. So Duchenne muscular dystrophy, G6PD deficiency, hemophilia, these are the disorders associated with X-linked recessive pattern, and they have no male to male transmission. Rather, they jump from one generation to another, skipping one generation, uh, and from mothers to son. So mothers are only carrier because they have two chromosomes, X chromosomes. If one will be abnormal, the other will compensate, and they will transmit to their sons. And mothers will be carriers, so one generation will be skipped and next generation will be affected. Hemophilia should not be confused with the example of von Willebrand autosomal dominant, and G6PD should also not be confused with it. And Duchenne muscular dystrophy shouldn't be confused with Becker muscular dystrophy. These are both X-linked, but they are a bit different, and not to be confused with the myotonic dystrophy, that is autosomal dominant one, not X-linked recessive. And then X-linked dominant one, if uh, a dominant disease is in the X chromosome, even present in any gender, it will be expressed. So there is transmitted through both parents, not from mothers, just like X recessive. Like it is transmitted from both parents. Any defective gene will be transmitted. Fragile X syndrome and Alport syndrome are two examples. They are asked. Hypophosphatemia rickets is not mostly asked. So S linked dominant one will behave like autosomal dominant one through both sexes, but it will be defective gene will be on X chromosome. Mitochondrial inheritance, we have read. Mila syndrome is important. And then coming on to the X-linked recessive disorders details, they are G6PD, hemophilia, Duchenne muscular dystrophy. We discussed only MCQ. Another MCQ is asked is Duchenne muscular dystrophy. Dystrophin gene is involved, the largest protein encoding human gene, and Gowers sign is positive because patient will stand up while uh, using his upper extremities to help stand up, and this is a Gowers sign. An MCQ is asked, Gowers sign is positive, it is Duchenne muscular dystrophy and it is X-linked recessive disorder. So male will be affected more, and in hemophilia scenario in G6PD, male patient will be given.